All right, couple housekeeping items. You may have already gone and taken a look at Moodle, and there's quite a number of things that are there. If we look at the syllabus, maybe, opening, there it is. All right. You will have two texts, the one that will finish up with the biosynthesis portion and then the molecular biology. When we transition, we learn lots of stuff, and specifically lots of stuff, and those are the topics of the stuff. Okay, this is what you really want to know. Okay. The breakdown, the exams 1 through 4 will be 125 points each. The final exam will be 150 points. There's a presentation, that's worth 100, integration of faith reflection, which is 100, and then between discussion quizzes and homework, there will be 150 points for a total of 1,000 points. We try to make it easy. And so in terms of grading scale, we tend to use the no plus minus in this class. All right. There are notes and things. There'll be drop boxes for assignments, and my lectures will be recorded and posted. So I'll put a playlist, I believe, under that Topics 4, so that you can go back and review the lectures if you need any help on that, or a review, or you missed something as you went through. All righty. Now, one thing that if you went through Biochem 2, I'm hoping you picked up some ideas about how to apply your organic chemistry. So we're going to start that with a review quiz. All right, let's shift gears just a little bit before we start to apply some of that knowledge. What do you know about fat? Made up of triglycerides and fatty acids are there, yes, very good. What else? Where do you find it in the body? Yes, the adipose tissue, at which the cellular type is totally different in terms of the types of enzymes that are there and regulation. And so that's different than muscle, which is what we normally talk about in biochemistry and in liver, so that it behaves differently to hormonal responses. What else do you know about fat? It's insoluble in water, yes. It's insoluble in water. So when fats are transported around and in the bloodstream from the tissue cells to mainly the muscle, it actually has to be carried by a protein. It just doesn't float around and it's not able to be dissolved. So there is some control in how we get that. In terms of energy, how would you classify fat versus the other energy source? Well, what are the other energy sources? You've got fat. What is it? There's glycogen in there. Yeah. So sugars, carbohydrates, fats, carbohydrates, and what's the other thing that we can break down for energy? Protein. Okay. If you compare fats, carbohydrates, and proteins, which one is more energy per gram? Fat. Okay. How in the world do we get energy from those fats, proteins, and carbohydrates? What's the process by which we extract energy and convert them from potential energy, the things we eat or the things we break down, into ATP? What's the general big picture item of that? Oxidation. We move electrons. And so the fats are very reduced. They're as reduced as possible. So we can do lots of oxidation all the way up to CO2. That means we can extract lots of electrons. And we extract those electrons in the form of NADH and FADH2. And that can be converted into a proton gradient, which then can be converted into ATP. And so fats are a large store of energy. Now, here in the American diet, a lot of people in the population eat more calories than they burn. okay? And it isn't always in the form of fat. They actually eat lots of carbohydrates. You can eat lots and lots of sugar and absolutely no fat and still gain fat mass, okay? And so if we do not use all of the energy that is incoming, the body tends to store that energy. And so we will go through the processes of our metabolism. And so things like glycolysis, citric acid cycle, and from those molecules we'll go into synthetic portions of metabolism. And so what we're going to do is we're going to see how we're synthesizing the fat from excess molecules that aren't being oxidized to use as energy sources. And so we will start with acetyl-CoA as basically the point at which we could branch towards the synthesis of fats in our body. And we'll first synthesize straight fatty acids, and then we'll synthesize some more complex types of organic molecules that are in the body. Now, acetyl-CoA, what stage or what pathway are we in when we talk about acetyl-CoA? It's really the beginning part of the citric acid cycle. Okay, the end of glycolysis is pyruvate, and the beginning of the citric acid cycle is acetyl-CoA reacting with the oxaloacetate. Okay, there is the pyruvate dehydrogenase complex, which creates acetyl-CoA. All right, that molecule could be sent to be degraded, but here, under the cellular conditions of the body, we're going to send it to synthesis. To do that, we're going to have a reaction that will make malonyl-CoA. Malonyl-CoA, it is what's called the committed step. What does committed step mean to you? Yep, it can't go back. It went down a road that can't go back to the other place that it has been before. Once you're at malonyl-CoA, you are now locking those carbons in the acetyl-CoA into a synthetic pathway. Okay? We're going to invest ATP, and what is this unit right here? What is that? Bicarbonate. It is an ion that we make GChem students memorize. Okay, but what does that represent in the body? That can be equated to something directly. CO2. It is CO2 that is reacted with water, which creates an acid that gets deprotonated to make bicarbonate. It is the buffering system of the biological system, and so when you see bicarbonate, there is an equilibrium between carbon dioxide and the bicarbonate, and depending upon how lazy the biochemist is who describes this, they could say CO2 in some books, they could see bicarbonate in other books, but they can interconvert very readily. Okay? And so, the reaction is carried out by 3 separate proteins. One protein is just a carrier in which it has the biotin molecule. Okay? Biotin is this particular molecule here. It is on a lysine, and what happens is it's a carrier. It'll actually take what you need from one active site in the carboxylase domain, and then it will transfer to a secondary active site. And so this carrier protein actually facilitates the reactions between 2 different active sites. The reactions go like this. The bicarbonate gets phosphorylated, and then the biotin reacts with that to place the CO2 on the nitrogen in the form of an acid. And then this CO2 gets transferred to the acetyl-CoA on the methyl group to create malonyl-CoA. Once I'm at malonyl-CoA, I have to go down synthetic pathways. Now, fatty acid synthesis. When we were talking about the acetyl-CoA, okay, what we actually are doing is we are putting a acetyl group on this very large organic molecule called CoA. And we sometimes talk about it. We show you the structure every so often, but this is the CoA part. There's an organic molecule, okay, in the middle here, that is the functional group. It actually includes that sulfur, okay? If you are attached to acetyl-CoA, or the acetyl groups, that's kind of the address that takes it normally to enzymes that do degradative processes, that break it down. And so there's something being carried, the acetyl group, that gets taken to enzymes that usually degrade. When we're doing synthesis, what we're going to transfer this to is something called APC. And it's a protein carrier of acyl units. Now what's important about this, it's on a small protein. So here you see you have serine. Attached to serine is an organic structure that looks just like CoA. So to the CoA's perspective, it's attached to the same thing. But now that organic molecule is attached to a protein, that takes it to the pathway for synthesis. And so if you have the CoA on it, degradative. If you have the ACP, synthetic. And so this is kind of the address. Now, if you take a look at the bottom picture, you can see the protein's is relatively small, but that organic molecule is relatively large. This CoA, even though we write only CoA, it is actually a very large cofactor. All right, there were 4 steps in degradation, so there are going to be 4 steps in synthesis. Can you remember what the 4 steps of degradation were? Hint, hint, it's kind of reversed. Okay, so what's the reverse of reduction? Oxidation. So we did an oxidation first, then hydration, and then oxidation, and then we did a really strange word that had lysis in it, which was break, but was lysis with a particular molecule, a particular atom, thiolysis. So to break it down, we did oxidation, hydration, oxidation, thiolysis. Here we go in reverse, and we actually reverse the steps. We condense, we reduce, we dehydrate, and then we reduce. All right, when we oxidize, what are the electron carriers that we use to oxidize? So if we do oxidation, the hydrides are accepted by 2 entities most often. Which are the 2? NAD plus accepts a hydride, yes, and the other is FAD. Okay? So reduction is done with a different molecule. What is that one? We've actually looked at it. We made it in the pentose phosphate pathway. Hint? NADPH. What is the difference between NADH and NADPH? Do you know? Is a reducing agent, yes. Structurally, what's the difference? Has a phosphate. I could have almost accepted NADPH as a P. Okay, yes, it does have a P because it has a phosphate. That one little change, the structure is very, very similar, but if one has no phosphate, it goes for oxidation. It accepts. If it has a phosphate on it, it's usually used for reduction. And so just small changes takes molecules to different places in the metabolism. All right, and so we have acetyl ACP instead of acetyl-CoA and malonyl ACP, and we're going to do a reaction to create aceto-acetyl ACP. All right, and so if you take a look at the leaving groups here, okay, we leave with CO2 and an ACP leaves. All right. And so if you do some inventory and you compare, All right, I've got to get something attached to something. Well, what is the things that are attaching? All right. Well, if I start thinking about it, when I actually do synthetic processes to attack, and I got lots of carbonyls, most of the time I'm thinking, I need a nucleophile. I need something negative to attack the carbonyl. All right? And I also need CO2 to fall off. All right, do you remember what CO2 looks like? Carbon, yes. Double bond to oxygen, yes. How many electron pairs are on the oxygens? How many lone pairs? 2. Come on, guys. My GChem students can answer that one. Okay, that's great. Perfect. If I look at what I have drawn here, I know I have 2 pairs on that oxygen. How many pairs do I have on the negative one? 3. All right. It almost looks like CO2, right? But it's not there. What would I have to do to make it look like CO2? You take one of the pairs of electrons and you can form the double bond. That's perfect. Now, what is the problem if I actually form that double bond? I'm making a new bond to carbon. Too many bonds to carbon. So one of them has to leave. Okay? I'm going to show you what the temporary intermediate is with a dotted line. I put the negative charge on a carbon. I make a carbanion. Is that thermodynamically stable? Usually carbanions are not thermodynamically stable. Okay? A negative charge on a carbon is not what wants to happen in organic chemistry. But I'm going to say that one is because why? Because of carbonyl. If there is a negative charge on that carbon that I just put it on, it can form a resonance structure and resonate into the oxygen. So that is actually a stable movement. Now, we're inside an enzyme, so we don't actually sit and have a negative charge sit there. Okay? What we do is we do something totally different because the reactant is right next to it. Wow, I just totally took everything away. Oh, I really took everything away. I got reverse happy. All right, I take this, make the double bond. This pair of electrons falls off and it becomes the nucleophile. Why does it attack that carbon? Well, one, we have a good leaving group. The protein is going to leave. Second, between the carbon and the oxygen is much, much more electronegative, so there's a partial negative on the oxygen, partial positive on the carbon, so nucleophiles like to attack. When I attack a carbonyl, what is the next step I must draw? The double bond of the pi must kick up and it forms a tetrahedral intermediate. That's the excited state. Then it comes down and then this falls off and picks up the proton. All right. I have condensed. I actually have put 2 of the molecules together. Now that doesn't look like fat right now. Okay? It's got a ways to go. All right, now we're going to do a reductase. And when you look at the reactants, you have 2 reactants. That ACP, the growing 4-carboned intermediate, and you have NADPH. Whenever you see NADPH, that means I can have a hydride attack. All right, will the hydride attack red or blue? Where would the hydride attack? At the red carbonyl or the blue carbonyl? The red. If you do inventory, ACP, ACP, sulfur, sulfur, ketone, ketone, no ketone, ketone. This is where the change happens. Okay? This happens inside an enzyme. It's a reductase. If you actually had this reaction done in an organic flask, like in the organic lab, the hydride could attack at either one of those, and most likely you're going to have attacks at both of those. And so if you actually did this reaction in the organic lab, you might get 50% of your product at best, okay? But in the enzyme, you're going to get one product 100% because the enzymes are directly where the reaction occurs. So for this, I always have a base and I always have a proton. In this, I don't lose any protons, so I'm not going to use a base, but I will have an extra proton. And so what happens is this hydride will attack, okay, and that hydride becomes that hydride. And then these electron pairs will pick up that proton, remembering the oxygen can be formally written as the negative charge goes on to the oxygen then picks up the proton, but it happens so quickly we draw that arrow. And so I have done a reductive step. So I condensed and I reduced. All right, the next reaction is to dehydrate. A water leaves. Okay, for water to leave, I have to pluck a proton off and I have to have a hydroxide leave. All right, and so if you do inventory, you can see no change, no change, carbonyl, no change. This carbon here, how many hydrogens? 1. How many here? 2. Okay. If I've got more hydrogens up here, that usually tells me exactly where to start. Okay, I found where I'm going to take my proton. And so in an enzyme, I always have a base. And I take that proton. Uh-oh. Okay, great. I took that proton away. What happens to that pair of electrons then? It will actually, it will form the double bond. Because I need to have a double bond in the reactant. Now I have to have a leaving group. What would the leaving group be? What's missing in the product? Water. And at that particular carbon, I'm missing the hydroxide. So the hydroxide can fall off, and it can become water. Now, what you don't see in the reaction is this. You don't see a reactant of a proton, okay? Because what happens net is I take a proton and I give a proton. So in the overall reaction, we don't write that because it's 1-to-1. And so, but water does come off as the final group. Okay, I've done condensing, I've done the reduction, I've done the dehydration. The last step is to do another reduction. All right. Red carbon. No, I'm not going to do that. Blue carbon. All right. You will have a proton as part of the reaction, and you will have a hydride attack. Now, where might the hydride attack? Is it the blue or is it the red? There is a reason why one is better than the other. Are we ready to vote? You have to vote. How many people say blue? I got 1 blue. How many say red? I've got almost the rest of the class red. Okay. Now, why in the world do you say red? Okay, I'm gonna attack that. I am going to do that because you told me to. Okay? I might get a bad grade, but I'm going to do it because you told me to. Okay? Why in the world, if you attack there, that's better? In that you can write the electrons can be on the blue carbon directly for a little tiny bit of time, which is alpha to a carbonyl, and alpha to a carbonyl can have that negative resonate, and so it's lower in energy. Now, it doesn't really sit there that long, it will then immediately pick up a proton, and this will be the proton, and this will be the hydride that came in. So the proton and the hydride is on the top, and the proton's on the bottom. I have created a fat. Woohoo! All right, a 4-carbon fat. You don't see 4-carbon fats too often. Okay, they're usually the degradative. And I haven't stopped. How long do we synthesize fats? The enzymes work and they'll continue to add 2 carbons at a time. When does it stop normally inside our bodies? How many carbons are normally found in a fatty acid? 16. Our enzymes are optimized to make 16. Okay? We would have to go through other processes to make longer ones, which longer ones are made and are important, but 16 is really usually what happens. Now, all of the enzymes that are described in this whole process, okay, there are 7 different active sites. Now, most of the experiments that are described in the chapter in biochemistry were investigated using bacteria, and what we actually discovered, there are 7 separate enzymes that were studied for this whole entire process. When we look at yeast, there are 2 proteins that still have 7 active sites, and in vertebrates, there is 1 very large protein that actually has 7 different active sites in its 1 long protein strand. And so, but these all work in concert. Whether it's the bacteria or the yeast, they do have to come into a complex to work. And the mammalian, the vertebrate, the complex is already formed as the protein folds itself. It is a cyclic process, and it will continue, and the enzymes are optimized to make 16 carbons long. In actuality, we not only work in a bundle of 7 active sites, but as you grow, you start with 2 on one, and then you make 4. And then when you do 6, actually, the 4 get transferred up to the other protein to make 6, and then transfer over to get 8, and it hops back and forth. But they can do 2 syntheses at a time. This seems to be, sugars are synthesized this way, fats are synthesized this way. There's something that is a motif in biochemical synthesis that we have dimers working together. Okay? Fat synthesis will take 8 acetyl-CoAs plus 7 ATPs plus 14 NADPHs plus 14 protons to make 1 16-carbon fatty acid. All right? Now, if you think about it in terms of energy, you have to use a lot of sugar to make those 14 NADPHs through the pentose phosphate pathway. Now, you made a whole lot more NADPHs at an exercise I did in Biochem 2, but it's a laborious thing. We are actually investing a lot of energy into this process. And so, but this molecule now actually has a very large store of energy, more than would be equivalent of 16 carbons in glucose, and it can be stored in a way that excludes water and is less weight than storing the same amount of energy in protein and or carbohydrates. There are some special cases. If you are going to synthesize fats that have odd numbers, you use propionyl ACP, a 3-carbon intermediate, to synthesize. We don't do that too often, but some animals do, particularly cows. We get odd fats from cow's milk and odd number fats. NADPH is used for the reduction. If you do elongate past 16, it actually happens in a different compartment and by other enzymes. And then we can unsaturate fats because this makes a fully saturated fat performed by other enzymes. And so what is the benefit of having an unsaturation? There is a benefit for having unsaturated fat. In the cell membrane, that's where we have a lot of the benefit, in that if it's all saturated, then if it gets into the membrane, they can become less fluid because they can make really great interactions. Now, when we put an unsaturation in, naturally, what kind of unsaturation? There is cis and there is trans. Which do we make internally? Cis. And so that cis makes a big bend, and so that loosens up the membrane fluidity, and so it tends to make them have a lower melting point, and so cis is more healthy. Trans, on the other hand, which you're not supposed to eat trans fats, or everybody says don't eat trans fats. The reason is you're unsaturated, but it still has and can compact like a saturated fat. But there's another case about the unsaturations that are important. Some derivatives of the fats that are unsaturated are hormones. And so we need the unsaturation for a whole different class of fats that we'll look at in at the end of this chapter. In terms of where fats are synthesized, we have fatty acid synthesis really in the cytosol. And so that's what we just looked at. We're also going to look at, just a little bit later, that's also where cholesterol is going to be synthesized. Okay? Now, if I want to make them longer, or I want them to have unsaturations, that occurs in the ER. Okay? And then the fats don't normally just sit by themselves. They're attached to different things, whether it be to the glycerol to make a full triacylglycerol, which is a fat molecule, or to be placed in the membrane. And so we'll talk about synthesis of membrane lipids that occurs also in the ER. And so knowing where the synthesis is important. Now, the synthesis of fats, okay, are made from acetyl-CoA, and that occurs in the cytosol. But when we take pyruvate and make acetyl-CoA, you told me it was part of the citric acid cycle, and that is true. But that citric acid cycle occurs inside the mitochondria. And so that acetyl-CoA that's inside the mitochondria usually is going to oxidative phosphorylation. But if I need to synthesize the fat, I've got to take that acetyl group and get it out into the cytosol. And to do that, we're going to use a shuttle. Okay? And so we're going to use the citrate pyruvate shuttle. Depending upon how it works, we can also make a little bit more NADPH using this process. And so normally we are making lots and lots of acetyl-CoA. Okay? Usually it will react with oxaloacetate, just like I said, and we make citrate. That's great. But there's a lot of regulation down this reaction, and so we can stop it going this direction. So if I stop the enzymes here, but I allow citrate synthase to continue, citrate builds up in a very high concentration, an unusually high concentration. That is when it is able to hit a concentration point where it starts to bind to a transporter, which transports citrate into the cytosol. Citrate is an unusual molecule in the cytosol. Citrate's really made in the mitochondria. So getting out there means that you have too many carbons coming into the citric acid cycle, and now we are destined to do some synthesis. In this case, we can break down citrate in its two components, the acetyl-CoA, where we need, and the oxaloacetate. But oxaloacetate needs to get back in. I can do that by either taking the malate, its conversion, which is a reaction that is the reverse of the citric acid cycle. The malate can come in, or the malate can be converted to provate, and provate already has a process to go in, and we can reconvert back to oxaloacetate. If we go from malate to provate, we also make an NADPH. And so that is an important step. And so a lot of times we talk about the citrate pyruvate shuttle, and that's using that final outtake. All right, the acetyl-CoA is used for the synthesis that we looked at. All right, so what I would like you to do is to work on those quizzes. So what you gave me was just bonus. This is the real quiz. And I want you to go through those reactions and work with your partners to do that, but make sure that you understand. I would like you to do this. First, attempt to do it as much on your own as possible, then get together and compare. And then you convince someone else that yours is right. If you're unsure, don't just take what they say and say, okay, that's right. Ask why. And if you ask why, then they have to explain it. And that will help them learn. And if they can explain it well to you, then you learn and everybody learns. And once you've done that, come to me. And then I'll help you learn and make sure that you're learning the right way. All right? Thank you, guys. Have a great day.